1. Functions of the primary cell wall are: structure support, controls rate and direction of growth, regulates diffusion, resists turgor pressure, protection, determines cell shape. 2. Typical primary cell wall composition is polysaccharides, proteins and enzymes, minerals, glycoproteins. 3. Cellulose is a polymer consisting of long, unbranched chains of D-glucose connected by beta-glycosidic linkages. 4. Hemicellulose consists of expansibility, cell-to-cell adhesion, cross-linkage and tethering, regulating yield threshold, nutrient supply. 5. Pectin consists of porosity, expansibility, elasticity diesterified Hg, Ca2 plus binding, resistance to mechanical stress, cell adhesion. 6. AGPs consists of may occur in the cell wall or be anchored to the cell membrane, cell identity, structural integrity, Ca2 plus regulation signal transduction, plasticity unidirectional deformation, desiccation tolerance, membrane integrity, tip growth. 7. Lignin is cross-linking phenolic precursors, highly heterogeneous polymer crosslink in diverse ways. Hardwoods equals coniferal and synapyl units. Softwoods equals coniferal units. 8. Chitin is long-chain polymer of acetylglucamine forming crystalline fibrils. Basically cellulose with one hydroxyl group on each monomer replaced with acetylamine. 9. Cell-to-cell interactions consists of connections between cells, markers of cellular identity, cell communication, involve membrane proteins and proteins secreted by cells. 10. ECM is composed of collagen, elastin, glycoproteins. 11. ECM can help coordinate the behavior of all cells in a particular tissue. 12. The ECM of some cells are attached to the plasma membrane by a third kind of glycoprotein called fibronectin. 13. Fibronectin molecules bind to ECM glycoproteins and proteins called integrins. 14. Adhesive junctions. Adhesive junctions appear to have been the first to evolve. Found in all animal species. 15. Adherens. Adherens junctions are found in animals ranging from jellyfish to vertebrates. Single-pass transmembrane protein. 16. Cadherens type 1 and 2. Type I will connect to another type L. Type O will join with another type LL. 17. Desmosomes. Desmosomes join adjacent cells. Support tissues against mechanical stress. 18. Tight junctions. Unique to vertebrates. Contain proteins called claudins. Occlude or block substances from passing between cells. 19. Septate junctions. Septate junctions are found in both invertebrates and vertebrates. Form a barrier that can seal off a sheet of cells. Proteins given different names in different systems. 20. Communicating junctions. Allow communication between cells by diffusion through small openings. Small molecules or ions to pass from one cell to the other. In animals called gap junctions. And plants called plasmodesmata. 21. Gap junctions. Found in invertebrates and vertebrates. 22. Plasmodesmata. Cytoplasmic connections that form across The touching plasma membranes are called plasmodesmata. The majority of living cells within a higher plant are connected to their neighbors by these junctions. Plasmodesmata function much like gap junctions in animal. 23. Gram-positive bacteria have a thicker wall. Stain a purple. 24. Gram-negative bacteria, more common, contain less peptidoglycan, do not retain purple-colored dye, can be stained with a red counterstain, appear dark pink. 25. S-layer, and some bacteria and archaea. Additional protein or glycoprotein layer forms a rigid paracrystalline surface. Functions often involve adhesion to surfaces or protection. 26. Loose connective tissues. Cells scattered within a matrix that contains a large amount of ground substance. 3 protein fibers, collagen, elastin, reticulin. Adipose cells important for nutrient storage. 27. Dense connective tissue. Less ground substance, contains tightly packed collagen fibers. It consists of 2 types, regular and irregular. The collagen fibers of dense regular connective tissue line up in parallel. 28. Special connective tissues. Has unique characteristics, cartilage, bone, blood. Has unique cells and matrices, lets them perform their specialized functions. 29. Cell signaling. Signal 1. Direct contact. Surface of a eukaryotic cell. Proteins. Carbohydrates. Lipids. Signal 2. Paracrine. Effects on cells in the immediate vicinity of the releasing cell. Signal 3. Endocrine. Signal molecule enters organism's circulatory system. Affect cells very distant from the releasing cell. Signal 4. Synaptic. Provide rapid communication with distant cells. 30. Phosphorylation is key in control of protein function. Signaling requires modification. Protein may be activated or deactivated by this. Common one is removal of phosphate group. 31. Protein kinases. Class of enzymes adds phosphate groups from ATP to protein. 32. Phosphatases. Class of enzymes that removes phosphate groups. 33. G-protein coupled receptors. Act indirectly on enzymes or ion channels with aid of a G-protein. G-protein inserted between the receptors and the enzyme. 34. Intracellular receptors. Can pass through the plasma membrane of the target cell. Some bind in cytoplasm, some bind in nucleus. 35. Steroid receptor action. Each of these receptors has 3 functional domains. 1. Hormone binding domain. 2. DNA binding domain. 3. Domain that can interact with coactivators to affect the level of gene transcription. 36. Specificity in the role of coactivators. Target cell response depends on the nature of the cell, even when different target cells have the same intracellular receptor. 37. Receptor tyrosine kinases influence. Cell cycle. Cell migration. Cell metabolism. Cell proliferation. 38. RTKs are activated by autophosphorylation. 39. RTKs are inactivated by internalization. 40. Protein kinase cascade. Series of protein kinases that phosphorylate each other in succession can amplify original signal. 41. 2 most common effector proteins. Adenylyl cyclase. Phospholipase C.